Okay, so I'm aware that the last 2 weeks I've given you quite a lot of difficult cell and molecular biology. So what I want to do this week is to try and build on that foundation that we've laid, use the hard work that you've all gone to in trying to understand this, to tell you a bit more about the research and the clinical trials that are ongoing. So, but the first 2 or 3 slides, I'm just going to try and go through what I've told you in order to try, because I think just by repetition helps it get through to you, and also to make sure that you appreciate the foundation that's going to be needed for what we're talking about today. So what I told you is that we start life as this single cell from your father's sperm and your mother's egg, and that builds up to make a whole human being. And that single cell has all the information in it to make a whole human being with all the different cells that we have. And that information is carried in our DNA molecule. And that's a fairly simple molecule. It's built of 4 building blocks, and I call them ATCG, but you can think of them as color. And there is a fairly simple rule that A can all pair with T and C can pair with G. But those building blocks are really arranged as beads, like beads on a string. So they're ordered, and this is what we call the strand, and these are the beads. But because then an A, for instance, or C can bind with a T, they can form this structure here, which is like a double strand, 2 strands held together by little bonds here, looking like a ladder. So it's a double-stranded ladder type structure that becomes twisted on itself, forming a helix, giving you the double-stranded DNA helix. At the other end of the scale, we have worker molecules, which are called proteins, and they have 20 building blocks, which are amino acids, roughly 20. And they also form on a linear string, but there can be strong interactions between them. And so they've become 3-dimensional in structure, and it's that precise shape and folding that enables them to function with the particular function they have. However, the information to make these proteins is in the DNA. However, and there's a factory called the protein factory, which will make the proteins, but it can't actually use this DNA information. So instead, a subcode is made, more or less resembling this, but subtly different, called messenger RNA, and that's got the code to make a single worker molecule. This, of course, has the code to make all the molecules that a cell is capable of making, which is something like 20,000. Messenger RNA is also built up similar 4 building blocks, but it's single strand like that rather than the double strand, and it's RNA. And I told you a little bit about how you go from DNA to RNA, and we're going to go through that a bit later again, so I won't reiterate that quite now. But what I told you is that these molecules are dispensable. They can be remade from the DNA, but what's really critical is that we maintain the integrity of the DNA. And by that, I mean not just an intact molecule, but also the sequence of these building blocks on that DNA, because it's that sequence that defines who we are as individuals. And that's what ATM does. It helps to maintain this DNA integrity. And I might add that AOA1 is functioning here as well. We talked about the inside of a cell being a dangerous place. It's subject to a lot of damage. All the molecules are damaged, but of course the critical one is maintaining the integrity of the DNA. We talked about reactive oxygen species, which are byproducts of our own metabolism and can cause a lot of lesions to the DNA, and this should be per cell, per DNA. They can be damages to those building blocks, those 4 different colored building blocks, or it can be damages to one of the strands, and sometimes to both strands at the same position, or even slightly overlapping positions. The most biologically significant damage are these, what we call double-strand DNA, double-strand breaks, i.e. breaks in both of those strands. And that's because the 2 molecules can then effectively come apart. And this is what ATM is responding to, a break in both of these DNA strands. And I mentioned also that these type of lesions, called double-strand breaks, can arise from ionizing radiation, because ionizing radiation is quite energetic. A track will pass through the cell and it can damage both strands at the same position. Sunlight, UV radiation, is less energetic and it does damage your DNA, but it doesn't damage both of the strands at the same position. So AT individuals have to be careful with ionizing radiation exposure, but don't have to worry about sunlight. So then we tried to think about what ATM is doing, and I likened it to a policeman rather than to being the doctor itself. So it's not actually repairing the break itself, it's facilitating the repair to be done in an optimal fashion. And it does this by sending signals when it senses that a double-strand break is present. AOA1, I might mention, is much more involved actually in the repair process itself. So what ATM can do is to tag, alter, add a little chemical group, which is called phosphorylation, to another protein. But it only does this to certain proteins, and those certain proteins have to have another tag that ATM recognizes. And it only does this when a double-strand break is present. So when double-strand break is sensed, ATM becomes activated and tags some of these proteins. It's able to tag about over 1,000 proteins, but in any one cell it won't do nearly as many as that. But it's this ability to affect a large number of processes by tagging these proteins that makes ATM have such broad manifestation. So it can stop processes taking place on the DNA. It says there's an accident down the road, stop more traffic coming down this road. We don't want to compound that accident. It can also activate a cell death pathway to remove very badly damaged cells. And the rationale here is that the cell doesn't want to be clogged up with very badly damaged cells, but also this is especially important for cancer, to remove cells that might otherwise become able to cause cancer. So ATM affects multiple pathways. It's activated by a double-strand break to tag other proteins and change their function in subtle ways. We talked, however, about what it's doing in terms of ataxia. So these known roles that a double-strand break can explain quite a lot, some of the features of AT individuals, such as the immune deficiency, the cancer predisposition, the radiation sensitivity. But the explanation for the reason of the ataxia is harder, because there are other individuals that are abnormal in the repair of DNA double-strand breaks, but those individuals tend to have small heads, but not progressive ataxia. And conversely, patients that do have defects in those single-strand breaks or abnormal transcription, they're the ones that have progressive ataxia. But the conundrum is that ATM is not activated by a single-strand break. And remember, that means a break in one of those strands. So the kind of potential explanations are that there's a lesion that accumulates because ATM is not helping to deal with it, that impedes the ability to transcribe And transcribe, if you remember, was making messenger RNA from DNA, and that's required to make new proteins. And because the Purkinje cells in the cerebellum are very transcriptionally active, they're making many proteins, they're metabolically active, they have high levels of these DNA damaging agents, they may be particularly reliant on ATM to help maintain their function. Okay, so that's the background to the last two talks, trying to summarize it as quickly as I could. But now what I want to do is talk about some of the ongoing clinical trials and also research approaches, but I'm going to be building and explaining to you in the terms of what we know about ATM. So the first one is some of the ongoing clinical trials, which are aimed at trying to delay the progression of ataxia. So one of the trials that you probably know about is called the Eridel trial, or is also called the ATEST trial. And that the rationale for that is that inflammation may be a factor contributing to the progressive ataxia. It may be a direct consequence of ATM loss, or something downstream, such as the fact that they're not functioning normally. And if it was this downstream function, then we would expect this treatment to be helpful for AOA1 patients as well. So the treatment uses dexamethasone, which is an anti-inflammatory agent, and you may have heard it talked about in terms of coronavirus, where patients, particularly those in hospital, are actually, their problems are compounded by severe inflammation, and dexamethasone can dampen that inflammatory reaction and help the patient survive. So again, the question is whether it's inflammation which is compounding, making worse some of the progressive ataxia. The critical aspect here is the delivery of dexamethasone in a manner which is slow. It's being delivered slowly, and that diminishes the side effects. So what's done is that the blood is taken from the individual, and the red blood cells or erythrocytes are infused with dexamethasone, and then that is put back into the individual and slowly releases dexamethasone into their bloodstream. It's called autologous because it's your own cells, and that diminishes any reaction against foreign material. Phase 2 trials have shown some improvement of the neurological impairment in 80 individuals, and so this is now a phase 3 trial. Just a couple of points. You always need controls in these type of trials, and the control here is that some individuals will be given a placebo and some the drug, but at the end of the trial, everybody will be given, even those on the placebo will be given the drug so they don't lose out. It's important that there's a careful and accurate scoring system for ataxia, and there's been quite a lot of discussion as to the optimum system. Obviously, that's, I should say that this is an international trial, so it is important that the scoring system is kind of done in an international way, so we're all assessing things in the same way. And it also needs very careful statistical analysis. That in these type of trials, it's not what's happening to the individual that matters. It's really, is there an impact on overall when you take the whole thing overall? And that actually requires quite careful statistical analysis. So my understanding is that the trial's now been completed. There's no more individual recruitment, but analysis is ongoing, and it may yet be a year before we actually get the results of whether this has been successful or not. Okay, so the other trial, another trial that I'm aware of is called IB1001. This is run by a company called IntraBio, and it's something called N-acetyl-L-leucine, or NALL. And this was used to treat vertigo patients originally. And basically, as far as I can understand, I explained to you last time when we were talking about neurons that the Purkinje cells and the cells in the cerebellum receive signals and transmit them. And that requires sort of the membranes to be intact and a potential across those membranes. So this drug has been used in a clinical trial for 2 disorders, but I'm just going to talk about this one called Niemann-Pick syndrome or disease, where the individual displayed progressive ataxia, dysarthria, dysphagia, and it had a positive impact in that clinical trial. It has also been used, so rather surprisingly, there seems to be a mouse model for this disorder, which is not the case for AT, because AT mice do not show progressive ataxia. But I think this is not really so much progressive ataxia as it's something called sigmoidal gait, and they have a sort of way of being able to assess that. And in these mice, they could see an improvement on treatment with this compound. This is something called a lysosomal storage disease, and basically the lysosomes are bodies that they have worker molecules, proteins, that will break down and help to remove and recycle larger molecules and sometimes waste products. And one of the impacts of this, so these lysosomes are not working normally in these individuals. And one of the consequences of that is that they have abnormal cell membranes, which I've said are important for neuronal signaling. And IB1001 seems to restore that in some way. It does, however, have a number of other impacts, and all of these could be impact. I'm not going to go through them individually, but these are all things that I've kind of talked a little bit about that could be impacting upon the ataxia. So the actual basis and rationale for improvement, both in this Niemann-Pick syndrome, is not completely clear, nor exactly how it might impact upon AT. But the good news is that it's easy to take. It obviously, you need careful assessment. So in this trial, you would have to go along to be carefully assessed, but it just involves taking medicine effectively. So it's not invasive or difficult to participate. Okay, another trial that I'm aware of that's undergoing. So what I should say, those 2 trials are both really based on compounds that are out there being used to treat other disorders. This particular clinical trial here is a bit more what I would call hypothesis driven. So it's based on learning about AT as a disorder, and therefore thinking about how you might exploit it. And the rationale is that, and it was kind of founded by Martin Lavin, who works in Brisbane and is an excellent scientist working on AT. And he found that there was this abnormal mitochondrial function in AT. So I had to explain to you what mitochondria are. So this is a cross section through the cell. This is the nucleus, which is where your chromosomes are, your DNA, most of your DNA is. And ATM is present here, although I think it has also been observed in the region outside the nucleus. This region outside the nucleus is called the cytoplasm. Just out of interest, these ribosomes are the protein factories, so they're where proteins are made. But then there's this little organelle, it's called a mitochondria. And the mitochondria basically are the powerhouses of the cell. It's where energy for the cell to do all its function is made. They also emit those reactive oxygen species, which, if you remember, were those agents that can cause damage to your DNA. I might just add that there is DNA in the mitochondria. It's different to the DNA I've talked about, and there are enzymes, there are worker molecules that can help repair and maintain the integrity of that DNA. But as far as I understand, ATM does not function in here. It's not found in this mitochondria. So this is slightly different. It's different to the inheritance, but I don't want to go into that. So there are patients with mitochondrial disorders, and they're pretty devastating disorders. And some of them at least show ataxia. And one of the possibilities is that's derived because there's not enough energy being generated, because this is what generates the energy, to carry out effectively that high transcription, making proteins, that metabolic activity that is required for the cerebellum. And what Martin Lavin was very astute and found was that cells lacking ATM, and more recently, not by Martin, but by others, I think this is true for AOA1 as well, have been shown to have abnormal mitochondria. Even though ATM is not in here, some of the consequences of not having ATM may still impact upon the mitochondria. They seem to have abnormal metabolism. So the normal route of making that power is via glucose metabolism. But they also emit high levels of these reactive oxygen species. So this drug here has been shown, because there's been quite a lot of research into how to treat some of these mitochondrial disorders, and it was able to treat the metabolic disorders primarily, I think, by replenishing intermediates that are required for that energy metabolism. So it allows the normal energy production. And my understanding is this is going to be tried in 80 individuals. Exactly where the trial is at, I don't know. I've just learned this from the web. But if anybody wants to know more, I could contact Martin Lavin. Okay, so they're in fact the 3 clinical trials that I'm aware of, although there may be others ongoing as well. Now, there's quite a lot of research going on into what is the basis causing that progressive ataxia in AT. And many are asking, some are asking, is it completely separate to the known function in that DNA damage response? And others are saying it is connected, it's likely to be connected to that, and I personally think it probably is, but what actual aspect of that damage response is ongoing? And some of these studies are being done in cells, some are being done in mice. There's a few where they're actually using aspects of the brain as well. I'm not going to go through all those types of studies, but I thought I would tell you this one for 2 reasons, partly because the AT Society had a grant call recently, and this is the grant, one of the grants that we've chosen to fund, and partly because it's slightly different to some of the others. And I think there is the possibility there, if this all proved to be correct, there would be the possibility for easy drug therapeutic intervention. So basically, the question here is, why are these Purkinje cells, these cells in the cerebellum, being lost with time? We're not losing other cells. There's something special about this region. And a factor in that may be these microglia, which are cells that are within that cerebellum, that region. And they're kind of like quality control cells. And basically, they're able to destroy non-functional neurons, which is where you can think of it as a quality control mechanism. Or in another kind of analogy, it's a bit like immune cells, which are able to destroy cells that are recognized as foreign. In this case, it's not so much they're being recognized as foreign as being recognized as non-functional. So the microglia normally would be sort of fairly what's called resting, but they are sensitive to signals that can come from external sources, but also can come from non-functional neurons. And those signals activate the microglia, and the microglia then send signals themselves to the neurons and cause them to die. So so the person doing this grant is Svetlana Korolenkova, who it's a Russian name, but she's based in Cambridge, and she's found that the microglia in cells lacking ATM are hyperactivated. And what she was able to do was to take, now I need just to explain to you a bit of terminology here. Wild type neurons mean normal neurons. Sometimes we call these normal neurons, but that implies that the AT neurons are abnormal, which is probably the case in this case, but it's not always the case. So wild type just means normal. And ATM here means ATM minus means you're not making any ATM protein. And this means, you remember I told you that ATM gene, the code, is on chromosome 11 and you have 2 copies. So this means both those copies are not there, or they're there, but they're not functional. So you're not making any ATM protein. And this is just kind of standard nomenclature. So what Svetlana did was take the microglia from cells which weren't making any ATM and mix them in a petri dish in the lab with neurons that were from wild type cells. And what she found was that the wild type neurons were destroyed by these microglia. So and then in another model system, it was found that if the microglia, the ATM minus microglia are inactivated, and there are drugs that will do that, then the ATM neurons persist. So in other words, it's suggesting that because these are hyperactive, they're destroying some good neurons. So in an AT, certainly the neurons won't be completely functional. They have some abnormalities, but they may not be that bad. They may be able to do some function if it wasn't for the fact that they're being destroyed. And this may explain why it is you're specifically losing these cells in the cerebellum. And certainly these cells, these microglia from ATM, appear capable of destroying good neurons, at least wild-type neurons. And the question then is, would this also be the case in AOA1? And it's hard to say, but it's certainly possible if the AOA1 neurons are sending signals that are activating all of this. So what Svetlana is going to do is look at this in more detail, trying to establish both the mechanism and the signaling mechanism to understand what's going on, but also consolidate that this might be the case. So trying to understand the mechanism behind this is important, but also we were kind of quite excited about it because it's always easier to dampen something that's hyperactivated. So there are definitely drugs out there where you can dampen this, make it not happen if it's really destroying good neurons. That's much easier than trying to get something to happen that, like, this wasn't functional at all. To get it to happen is much easier to do it. So the chance of it being exploited therapeutically is definitely there. Okay, so there are some of the kind of more research aspects. What I'm going to talk to you now is some approaches to try and get that ATM protein expressed in ATM neurons. So what we call delivery of ATM to neurons. And then I'll come to other approaches that all fall into this general category. So what do we mean by delivery? And basically, what we want to do is have that wild ATM, remember I called it the worker molecule, the protein, doing the job in neurons. And wild type means the normal ATM protein. Now you'd think what you might be able to do is make ATM protein in the laboratory and give it as a tablet, and we'd be able to get it into our cells. Unfortunately, that approach is not really possible. Firstly, ATM is very large protein. We haven't even been able to make it actually in the cell, but certainly not in a way that you could package it into material. But secondly, it's got to get through to the neurons, and that is particularly difficult to do. So the approach then would be, so let's just go back to this. I talked to you about DNA, your messenger RNA, and your protein. And remember what I told you is that your messenger RNA is not a contiguous piece of DNA that's on your DNA, or contiguous sequence that's on your DNA, but instead of this, there's this splicing where you cut out bits that are then removed. And this, if you remember, I was calling splicing. So your messenger RNA is much shorter than the region on the DNA, which is called the ATM gene. So remember, this part, being able to make the protein from the message, is all functioning in an AT individual. The problem is this code is subtly changed. So, and now already the ATM protein is very large. This is large, and this is even larger. And there have been problems because this has about 8,000 building blocks to it. So we definitely don't want to be trying to put all that back into the cell. It's just too large to work with. But the trouble is that RNA, messenger RNA particularly, is not very stable. So instead of trying to put this back into the cell, we put back, what we do is take this in a laboratory, and we've got the means to be able to reverse this code and go back to this, but we're not going back to this with these redundant bits of information. We're just making DNA that is this exact copy of this messenger RNA. So what we want to do is deliver that into the neurons, and that will then make the messenger RNA, which will then make the protein. Now, and that, so unfortunately, if we just give the cell cDNA, there are molecules, worker molecules in the cell that will destroy it. So what we try to do is give it to a carrier that will carry this ATM cDNA and allow it to enter cells. And often in this type of approach, what you use is a vector, a virus vector. So basically, think of the coronavirus. It can get into cells. It recognizes what are called receptors, but things on the surface of the cell, grabs onto them and uses that to get inside the cell. And so we want to exploit a virus to be able to do that and help us get this bit of DNA. And we can put the bit of DNA into the virus so it's carrying it. That bit's quite easy to do. And then exploit it to get it into the cell. Obviously, we don't want 80 individuals to become infected with whatever the virus is, certainly not coronavirus, but it's actually quite easy to disable the ability of the virus to replicate. That's not a problem, it's been well done. So the problems are the ATM cDNA, I just told you about, is very large, and it's been too large for many vectors. But in fact, now I think that's largely been overcome. We've identified vectors that will carry that larger ATM cDNA. The one challenge is the blood-brain barrier makes it difficult to get this, which is a barrier allowing larger molecules to get into the brain. That remains a challenge, but there are ways around that now. But the major challenge has still been to infect neurons, and particularly the Purkinje cells. So in the recent grant call that we had, though, just to give you an idea, we had 14 grants, and I think 4 of them were all sort of variations on this approach. So there's quite a lot of work going on in this. But one of the grants, which was a very nice approach, used something called adenovirus, which is actually the same virus that's used to carry the spike protein in order to be able to make the vaccines. So, and that's been given to loads and loads of people, so it's perfectly safe. That's the AstraZeneca virus, I think. But it's called helper dependent in this case, and that's done because it's got specific receptors on it that will recognize the neuronal cells. But the trouble is, it doesn't seem to be recognizing the Purkinje cells well. So there's the proposal was to change it, and it was very elegant work to try and make it recognize and select the cells that can get into the Purkinje cells. It's still difficult to deliver it because of this blood-brain barrier, and there's various different ways that aim to overcome that. And there's still a bit of a challenge for how much do we need delivered? Does it have to affect every single cell, Purkinje cell, or would affecting 10% be enough? And how stable is it? How long will that protein worker molecule be made even if we infect the cell? So this is ongoing work, but I think progress is definitely being made here. And there's also some other work that the AT Society has funded, where we've used a non-viral vector system to deliver this ATM cDNA. So it's very similar, but it's just a different delivery system. And that work's progressing well too. Okay, so that's what we call, in a way, vectorology. It's how we're delivering the means for the cell to start making ATM protein again. Now, a second kind of approach here is correcting the mutation that's in the cell. And I'm going through this approach first, and then I'll do another one where you're actually changing that mutation. And this is an approach that's been promoted by the AT Children's Project in the US. Now, you may remember, and I'm not going to go through this in detail, that we talked about different mutations in AT individuals and what we meant by mutations, which is a change in your DNA sequence. And that's the fundamental difference that's happened in your children and in carriers as well, that you've got a change in that sequence of the building blocks that changes that sequence in your DNA, and that change gets translated or gets converted into a change in your messenger RNA. In this fairly simple type of change, we just have this one the one building block here change from a T to an A. And because, remember, I told you the code is read in groups of 3, that changes a building block in the protein from cysteine, in this case, to a serine. And it's that single little change in that building block in your protein that can make the protein fold completely different. So that was a missense mutation. I also explained that you can have mutations. It knows where to start and where to stop, and that sometimes you can get a stop mutation and sometimes a frame shift mutation. And I'm not going, this was all explained in lecture 1, and I'm not going through that again, but what I want to focus on the 4th group here was called splicing mutations. And if you remember, I told you can have a sequence on your DNA, and this may be within the ATM gene. And then you want to make your messenger RNA. But you don't, although all the information for this messenger RNA is in your DNA, it's not contiguous. And there are little regions that are just thrown away. And we call these non-coding or intron regions. And they're not providing the information to assemble your protein. And for ATM, I said there's 66 times that you make these cuts, throw this bit away and join them together. And some mutations can arise because you don't do this at the right position. And these are called splicing mutations. And it's quite complex how the cell knows where to do this cut and splicing. It's not a definite little sequence here. It seems to be, there are what we call consensus sequences that are preferentially used, and but it's quite complex. I've shown here these just being, I don't know what I've written here, 10 or 15 little building blocks, but actually often it's more like 100, 200 or even more. So these things here are quite large. Now, splicing mutations, because of the complexity, are actually quite common in AT. My understanding is that they can contribute to up to 50% of the mutations that arise in individuals. But this approach that the AT Children's Project are using is only going to be suitable for a small subset of individuals. And it's those individuals where the mutation has created a new splice site. And so normally you might splice from there, you would be splicing from there to there. Here you might go from there to there, because this is now recognized as a splice site because of the mutation change. And then you might go from there to there, and whatever that will give you these additional sequences in the code, which could be disastrous. So what this approach does is puts in something which is called an antisense oligonucleotide. And basically, this is a little sequence of building blocks that are able to bind to and recognize this sequence here. And because this is being done, this is the pre-messenger RNA, so it's before you've done this cut and splicing. This is single-stranded. So if you give it this little sequence, it will very avidly go and bind here. And by binding, it can prevent this being used as a splice site. So then you get normal restoration. Now, of all the splicing mutations, there's not so many that arise from creating a new splice site here. So at the moment, this approach will only be useful for a small number of individuals. Nevertheless, and this is called antisense oligo because it's an oligo just means a short stretch of DNA, but antisense means it's the A. I told you the A always binds with T, so this would, if you want the sequence here to be binding, it would be T, T, T. C always binds with G. So it's the opposite of it that will enable it to bind to it, if you understand. So a patient, as I said, has been identified, a young patient. I think there may be more than one, and they've taken cells from that patient and they've been able to do all of this in the test in the laboratory, and works, and they can restore the radiation sensitivity of the cells, and they can see ATM is being made. And now they're trying to do this in the clinic. So they're giving this individual these antisense oligonucleotides and trying to see if there's any improvement on the ataxia. It's a young patient, so a young girl, I think. So we'll be able to see whether it's progressing, whether the ataxia progresses normally or not. So currently, this is only applicable to a small number and will be only beneficial to a very small number of individuals. But exactly whether it could be progressed for other types of mutations, we have to wait and see. I think it's very important, and for two reasons. One, it will enable us to see whether, if you can treat a person after they're born, you can stop or even send back the progressive ataxia. That will be hugely important just as proof of principle. The second thing is, I think it's exciting technology because these are not large molecules. They're short oligonucleotides. And so, although we have to work out further ways to see how we can exploit this, it could still be very important to be able to see that we might be able to make some changes. Okay, so in a way, this approach is trying to get your ATM protein to be made in the cell. It's not actually changing the mutation itself. That change will still be in your DNA, but it's intercepting between the DNA to making the protein. This next approach is subtly different in that it's editing that sequence in your DNA, and it would be an aim to restore a normal DNA sequence. And CRISPR is a technique for trying to change DNA sequences. And this has gone apace in the last few years. And last year there was a Nobel Prize awarded for this technique. And I don't want to go into this in detail, it's quite complicated, but it basically came from work with bacteria, where they found it was a system that bacteria use to ward off infections, basically. But it's been adapted to using in human cells, and it uses something called the Cas9 protein, which is able to introduce double-strand breaks into your DNA. But where it introduces them is geared by something called sequence-guided RNAs. And basically, for any region of your DNA, it can make one of those little, almost oligonucleotides almost, to gear anything to go and bind. And so this little sequence here will recognize the sequence in your DNA. And that means you can make it go to any position in your DNA that you want, which is a huge step forward. The traditional technique has then been you activate these double-strand breaks, but that's not very good, because then if they're misrejoined, that creates a mutation. And that's been used to create mutations. But we want to do for 88 individuals is change your sequence here. And I don't want to go into this in detail because it's quite complicated, but there is technology emerging to try and change some of this, the sequence, so as to make it back to the sequence that it should be. There's bridges to cross, it's got to get into the neurons, but it's an approach that's being used with muscular dystrophy and looking quite positive at the moment. But that's much easier because you only have to get it into the muscles. But so we're not there yet, but there is hope on the horizon. So that's really what I'm going to talk about today. There are these other areas that I think are still important areas for research and important areas to be telling you about. So I'll try and cover those next week. And based on the talk we had earlier this week, I'm going to also talk about the cancer predisposition in AT, and we'll mention about the immune deficiency. Okay, so